Hi everyone, today's topic is ELISA. Before starting the topic, like and subscribe my YouTube channel. What is ELISA? ELISA is the basic assay technique known as enzyme linked immunoabsorbent assay, also referred to as EIA, enzyme immunoassay, that is carried out to detect and measure antibodies, hormones, peptides, and proteins in blood. Antibodies are blood proteins produced in response to a specific antigen. It helps to examine the presence of antibodies in the body in case of certain infectious diseases. ELISA is the distinguished analysis compared to other antibody assays as it yields quantitative results and separation of non-specific and specific interactions that take place through serial binding to solid surfaces, which is normally a polystyrene multi-well plate. Next, types of ELISA. ELISA tests can be classified into three types depending upon the different methods used for binding between antigen and antibodies. There are three main types which are named as indirect ELISA, sandwich ELISA, competitive ELISA. Indirect ELISA. In this, antigen is coated to microtiter well. Next, sandwich ELISA. In this, antibody is coated on the microtiter well. Competitive ELISA. In this microtiter well, which is antigen coated, is filled with the antigen antibody mixture. Indirect ELISA. Indirect ELISA detects the presence of an antibody in a sample. The antigen is attached to the walls of the microtiter plate. A sample containing the antibodies is added to the antigen coated walls for binding with antigen. The free primary antibodies are washed away and the antigen antibody complex is detected by adding a secondary antibody conjugated with an enzyme that can bind with primary antibody. All the free secondary antibodies are washed away. A specific substrate is added which gives a colored product. The absorbance of the colored product is measured by spectrophotometry. Second, sandwich ELISA. Sandwich ELISA helps to detect the presence of antigen in a sample. The microtiter well is coated by the antibody. The sample containing the antigen is added to the well and washed to remove free antigens. Then an enzyme linked secondary antibody which binds to another epitope on the antigen is added. The well is washed to remove any free secondary antibodies. The enzyme specific substrate is added to the plate to form a colored product which can be measured. Competitive ELISA. Competitive ELISA helps to detect antigen concentration in a sample. The microtiter wells are coated with the antigen. Antibodies are incubated in a solution having the antigen. The solution of the antigen antibody complex is added to the microtiter wells. The well is then washed to remove any unbound antibodies. More the concentration of antigen in the sample, lesser the free antibodies available to interact with the antigen, which is coated in the well. The enzyme linked secondary antibody is added to detect the number of primary antibodies present in the well. The concentration is then determined by spectrophotometry. So it was all about a short introduction and types of ELISA.